Well, good morning, everyone. We're glad to have you join with us today for our Texas Corn Producers Board first installment of Corn Conversations. I'm David Gibson, the Executive Director for the Texas Corn Producers Board, and we serve the state's farmers through two organizations here in our office. One is the board funded by checkoff monies, and through our association, Texas Corn Producers Association, funded by corn farmer memberships. During today's event, we're going to share some exciting developments in fumonisin mitigation that are made possible through our checkoff efforts. This goes back to some real severe issues in 2017 and some issues we've seen over the years with fumonisin. A few things I want to walk you through before we start the conversation. This is a webinar format. We are unable to see or hear those participating. We will have time for questions following the speaker's presentation. To submit a question through the Zoom app, you may press the Q&A button at the bottom of the screen. You'll then type in your question. If you are participating by phone audio only, you may submit questions via email to cornconvo@texascorn.org, CORNCONVO at TEXASCORN.org, or call our office 806-763-2676, and someone will answer the phone and take your question. This conversation is being recorded and will be made available on our website at texascorn.org. Here to introduce today's speaker is Charles Ring. Charles is a farmer from Sinton, Texas, and he's chairman of our Texas Corn Producers Board Research Committee. Charles.
Good morning. Research is a key part of the efforts of our state checkoff and supports on behalf of Texas corn farmers. I'd like to explain that this research initially came about in that the question came up, okay, can we use a biological control such as we have for aflatoxin with Afligard and AF36 Prevail now, that we can apply and get some benefit and a tool, to go into prevention. It was a little surprising as to what's been discovered, and that's what we will talk about today. And I'm glad to introduce Dr. Ken Obasa, a plant pathologist with Texas A&M AgriLife Extension in Amarillo. He's among the many researchers working on advancements for our commodity. In 2019, Dr. Obasa began studying the efforts of bacteria to control fumonisin levels in corn in Texas, Colorado, and Kansas. The study is made possible through a partnership led by Texas Corn Producers Board in conjunction with Colorado Corn Administrative Committee, Kansas Corn Commission, and the National Corn Growers Association. The project is intended to find a new, affordable tool to mitigate the prevalence and concentrations of fumonisin in grains. Thank you for joining us, Dr. Obasa, and we look forward to hearing from you. Go ahead.
Thank you, Charles. Can you hear me? Thank you, David. Thank you, everybody, for joining us today to be a part of this webinar. My name is Ken Obasa, like I've been introduced, and I will be sharing with you findings from the work that I've done that Charles just described, looking for possibilities, microbial possibilities among bacteria that can be used as potential biocontrol agents for the mitigation, management of fumonisin in corn. And so the project really is titled officially The Survey of Cornfields in the Texas Panhandle and South Plains Kansas and Colorado State for bacteria with potential for the management of fumonisin in corn. And the project really has 4 key objectives, okay? The first is obviously to collect the corn plants from selected counties in those 3 states of Texas, Kansas, and Colorado, and then bring those back to our facility here in Amarillo in Texas and try to isolate any bacteria that might be living and naturally occurring within those corn plants that we've collected. Once those bacteria have been isolated, we will then evaluate and identify those bacteria for their ability or otherwise for benefits such as preventing the fungus from being able to grow, and their ability to prevent the fungus from making fumonisin, or maybe reducing the fumonisin level that the fungus is able to make naturally. The last part of it is, if we find candidates that show promise along these 2 criteria, we will then proceed and begin to sequence to try to identify genes within this bacteria that's making these abilities possible, so we know exactly the mechanism by which they're doing this. All of these objectives fall actually under the fumonisin and integrated pest management priority area of the Texas Corn Producer Board for 2019, which itself falls under the production and management category of that priority area. So as a way, by way of introduction, you all probably know this already, but the Texas Panhandle and South Plains are actually 2 very important districts in the state of Texas. In 2017, and it follows through in 2018, that both districts actually represented about 70.5 and 18.1% respectively of the state's total agricultural production. And in the same 2 districts, you have the top corn production districts in Texas represented there. And then if you look further into that, you find that the top 3 production counties in the state of Texas are actually in District 1, north of the Canadian River, so Atlee, Dallam, and Sherman, respectively. And connected to this, this is a plant business we're doing here today, but agriculture is all interconnected. And right now, about 25% of the beef supply to the US market actually comes from District 1 here in Texas. And of course, these animals have to eat, and corn is part of what they eat. So this all kind of ties together, making this project really significant. Next, I kind of want to touch on the rationale that went into designing this project and the approach that we're taking. And it really ties to 3 things that I kind of want to emphasize especially. And the first is the recent and ongoing variability in weather in Texas that we've been experiencing lately, especially with respect to heat and drought. These 2 have the potential to exacerbate the cost of corn production. They can also lead to increase in incidences and activity levels of mycotoxigenic corn fungal pathogens like Fusarium, which makes fumonisin. As of 2 months ago, there's a report coming out from Canada, because of increasing temperatures. They've been reporting increasing levels of fumonisin as a result of increased infection from Fusarium fungal pathogens on their crop. So that's already becoming, like a thing that's, being experienced in Canada. The other thing is, it could lead to increases as a result of higher incidences of this fungus. You could have higher levels of mycotoxin contamination resulting from that in corn. Overall, this could lead to reduction in corn production in Panhandle and South Plains, and consequently reduce economic returns for corn production here in the state of Texas. The second rationale that kind of went into this is non-weather related, and it kind of comes from the angle of the pathogen itself that makes this fumonisin, okay? And so recently we've been identifying, there's been increased cases of the identification of, a species of this fungus, Fusarium verticilloides. It actually also infects corn, and it, contaminates it with fumonisin. But increasingly we've been finding cases of strains of this fungus that are actually resistant to the DMI class of fungicide, so the methylation inhibitor. So I'm talking about your triazoles, your imidazoles, so kind of propiconazole, tebuconazole, and the likes of those. So now we're finding strains of this fungus that are resistant to these fungicides. So when you apply them, either you don't get control or you don't get as good a control as you should, okay? The other thing is we're finding also increased incidences of another species of the same fungus, Fusarium, called Fusarium proliferatum in corn. So naturally, these fungus, this very species proliferatum, coexist with Verticilloides. They are frequently isolated from infected corn, different species, but the increase in the incidences and isolation of this particular species, proliferatum, is of a little concern. And for two reasons. It's because the group that falls under that species are among the highest fumonisin producers in the Fusarium genus. And this particular species doesn't make just fumonisin, it makes other mycotoxins as well. So buvericin, fusaproliferin, and moniliformin. So these are important mycotoxins as well. So the potential here is that, the case of fumonisin can then be compounded, by all these other additional mycotoxins that this particular species is able to make. But you can contrast all of that issue that I just described here with respect to, findings in the fungus, the resistance, and, the one that makes multiple mycotoxins. You can contrast all of that with, what we know as of right now when it comes to the use of biologicals, biocontrol agents, like bacteria, for the management of, disease in corn. As of yet, there has been no record of resistance evolving against the use of biologicals for managing the pathogen that produces mycotoxin. And so that's promising. It makes this approach at trying to mitigate this fungus one that's worthwhile looking at and investing in. The last rationale that I want to talk on real quickly is recent discovery. So our understanding, we continue to evolve in our understanding of what we know about the pathosystem, how pathogens and microbes interact with plants and the environment. But of recent, there's been some new discoveries. So areas of plant pathology that we've not really looked at for the longest time. Well, basically what we're finding is that in nature, whereas we've always thought about fungi as being fungi, like they exist by themselves. It's just a fungus. But in reality, it turns out that they don't actually exist by themselves. They're what we term holobionts. And what that means basically is that an organism that has another organism living inside of it. Now, this organism that lives inside of this fungus, the endosymbionts, as we term them, they're not just, idling away doing nothing there. It turns out that they actually impact the lifestyle of the fungus or their host. The areas that I really want to emphasize here is that we're finding now evidence that the presence or absence of this bacteria within their host fungus actually has effect on the ability of this fungus in some cases to cause disease or how much disease they can cause. And in some other cases, we found instances where, like the presence or absence actually affects the ability to make secondary metabolites, like mycotoxins or virulence factors. And in some other instances, we found that, without this bacteria, the fungus is actually not able to reproduce. Now, that's actually very important because when you think about, what goes on in the field, an initial infection comes in and then it multiplies and spreads in the field. That spread is made possible because the fungus is able to reproduce. So to think that, like there is a potential way to actually stop the fungus from reproducing opens the possibility for being able to control the spread of the fungus in the field. And this particular last rationale, to further illustrate them, I want to talk on two specific examples to kind of make my point. And the first is a work that was published, myself and some groups from my colleagues at the University of Florida, Kansas State University, and the University of Kansas. And this was work done in Rhizoctonia solani. And this is also a pathogen that goes to corn, okay? And so basically what we found for the first time is, if you can see my cursor here, and for those who are just audio only and listening in, basically what we found is that we were able to show that, there is this bacteria living within the fungus by DNA probe. We probed for the DNA of bacteria, we showed that. And when we take that natural native fungus and we treat it with antibiotic, you can actually remove all the bacteria from inside the fungus. And that's, for those who can see, that's what this area here is. So this is 2 of them, and then we repeat them in duplicate, so there's 4, and after treating them with antibiotic, you don't see any DNA like you see here. So it means we can get rid of this bacteria, okay? And then another thing we did, which is shown here, so all of this, first of all, is showing you evidence that, like bacteria do live inside of fungus, okay? So this slide, for those who can see, we're showing you the nucleus of the fungus, so this bright green blobs here are the nucleus of the fungus. And you've got, the one we've treated with antibiotic, you don't see anything here. This is electron micrograph, okay? And again, to make the point, we did electron microscopy, and we're showing, so all this, white round blobs that you see inside of these are the bacteria. They're actually inside these thread-like structures are actually the fungus, okay? And we did the same thing again by, making the bacteria to glow green, so we transform them so that they can express a green color. So every time you see the bacteria, it's going to be glowing green. And again, within the fungus, you can see the bacteria here, okay? So this was our first evidence that, hey, this fungus, even though we've been working on it for over 150 years, has always had bacteria living inside of it. And so, what exactly is this fungus then? But what does this association mean for the biology of the fungus? That's where it gets interesting, okay? And, without going into much details, I'll just kind of summarize the slide you're looking at for those who have the opportunity to see what I'm presenting here. And for those of you that are just audio only, I'll try to summarize it in a way that you'll understand. And so basically what this slide is showing, the top one here with the pot and the grass in there, so this is creeping bent grass, it's a tough grass, is, so this is the tough grass. We did not inoculate or introduce anything to it, just the tough grass by itself. It's growing fine, it's green. The next one is the same grass, but to this one, we introduced the fungus that we also had treated with antibiotic. So now this fungus no longer has the bacteria, okay? And you can see there's a lot of green tissues here. It's looking fine, it's not sick or anything. But although the image can't show it much, the fungus is growing there, but it's not causing disease, okay? Now, the third part you're looking at is one where we introduced the fungus, okay? And we didn't treat it with antibiotic, okay? And we didn't do anything to the bacteria. So this is the native fungus. And you can see it just kills this grass, okay? Now, this last part that you're looking at is one where we take the fungus that we had used in this second part that had the fungus that had been treated with antibiotic, and so it wasn't causing disease. Well, after treating it with antibiotic, we put the bacteria back in there. And again, you see it kills the plant. So basically, anytime the fungus has the bacteria within itself, it's pathogenic. It can cause disease. But we looked further, and what we basically found is, if you look at this fungus, in addition to being able to cause disease, it makes a secondary metabolite that actually allows it to be able to cause disease, okay? That thing is, the compound is phenylacetic acid, PAA. And so on the x-axis, you have the amount of PAA that's been made. And so this one where you have the yellow arrow on right now is the natural fungus. You can see how much PAA it's making. Contrast that to this, okay, where we have treated the fungus with antibiotic to eliminate the bacteria from within it. Suddenly, it's making a low level of phenylacetic acid, PAA. But when we put the bacteria back into it, the third one, see, the amount of PAA suddenly jumps up again, okay? That is one case. The second example I want to use to make my point here is this other work that we did, and this was just published in January of this year. Myself, colleagues at University of Nebraska, University of Florida, and University of Kansas. We did this work, and this was in Fusarium. Okay, so now we're coming closer home to corn, even though Rhizoctonia goes to corn as well. And what we're finding basically is that, well, it's kind of, like analogous to what we found with Rhizoctonia in the sense that, Fusarium turns out also to have bacteria living inside of it, okay? And just for sake of time, a few of the things we found highlight is, hey, in the absence of this bacteria, it looks like this fungus is not able to make one of two kinds of spores by which it spreads in the field, like I described earlier. It's not able to make one of those two kinds of spores. And so here we have the bar chart on the left is the first type of spore, which is called Microconidia. Okay, so you have the yellow arrow right now sitting over the bar for the wild type, the native natural fungus. This is how much spores it's making in one meal. You can make up to 30,000 of that spores. Well, after treating that same fungus with antibiotic, it makes 0 of that spore. Okay, when you look at the second type of spore that this fungus is able to make, the Microconidia, okay, well, unlike the Microconidia, it's still able to make, a good amount of that. But if you contrast that with what those strains of the same fungus that has the bacteria in them makes, it's much higher than what the one without the bacteria makes, okay? The other thing we found in this particular case with the Fusarium study is that, the symbiotic bacteria that are associated with this fungus actually affect the ability of the fungus to make fumonisin, which is really, what this project is all about, okay? And so it turns out that if you look on the bar chart on the left, the green arrows right now are sitting on what should be the spot where the bars should be for the fungus that have been treated with antibiotic. But you see that the amount of fumonisin that they're making is 0. You can contrast that with the rest of the bars on the same bar chart on the left, and you'll find that, all of those bar charts actually represent a fungus that has the bacteria inside of it. They're all making fumonisin. The 3 different classes of fumonisin, fumonisin B1, B2, B3 is what we looked at, okay? Now, what we did next was we took all of those ones on the left bar chart that actually make fumonisin, except for the wild type, the native one. We treated all of those with antibiotic 2 and C just to see if we can eliminate, and reproduce that same thing. And if you look on the bar chart on the right, so on the left, they're making fumonisin, and suddenly on the right, it's all down to about 0, okay? But this was even more interesting because we found out that all of these observations weren't just, happenstance. It was actually correlated with what was happening at the genetic level. So when an organism makes a protein or anything, hormone, they're actually the product of genes that are present within the cells of a living tissue. So for instance, a person who's got diabetes needs to make insulin. If the gene responsible for making insulin is not working well and not making enough, well, then they're going to need insulin administered externally from the outside. Well, so we looked at the genes, and I'm only showing 2 of those important genes that are necessary for the production of fumonisin. There's a cluster of them. It's not just one gene. There are close to 10 of them. When we looked at, the key genes that must, even if some of the others are not functioning optimally, these ones must function optimally in order for fumonisin to be produced. When we looked at 2 key ones, FOM1 and FOM21, it was interesting because what we found was in every instance where the wild type fungus was looked at, those genes were expressing at a high level. But when we contrasted that with instances where, we had treated that same fungus with antibiotic, the gene level was almost down to 0 or close to 0, which correlates with what we observed earlier that, hey, without this bacteria, we're not getting a whole bunch of fumonisin being made by this fungus, okay? So you think about all of that and you begin to ask yourself, well, if there are naturally occurring bacteria that can, for better or for worse, affect the ability of the fungus in certain of its lifestyle, is there a possibility that there are bacteria out there that have the natural potential to actually suppress the ability of this fungus to make fumonisin? In the examples I just talked about, the bacteria actually helps the fungus make fumonisin. But the target of this project is to find those that will suppress the ability of the fungus to make fumonisin. And so thinking of all of that, you kind of have to look at this project by the corn board as one that's a forward-looking investment to protect and enable the continued production of high value and healthy corn in Texas, as well as in Kansas and Colorado. The slide I'm showing you right now, if you can see it, shows some of the points, the survey points, locations we collected samples from in Texas, District 1 and District 2, as well as in Colorado and Kansas State. The next slide is actually a schematic of the workflow that we followed, that we implemented in carrying out this work, looking for those potential beneficial bacteria. And naturally, it starts with, collecting the corn samples from the field. And after that, we bring it to the lab and process, try to isolate any bacteria that might naturally occur within the corn plants that we isolated. So this is native. It's not like we're introducing anything. There is no, GMO here. We're just looking at what nature has to offer. And so we put those on a media that naturally favors the growth of bacteria and try to attract the bacteria to grow out of the corn stalks and onto that plate that you're looking at. And then, like you can see, if you can actually see this presentation right now, we recovered bacteria just like the ones you're seeing from those corn stalks, okay? And although it's not that clear, what we found, one of the things we found is, from the corn stalks, you would think, oh, maybe one, maybe two kinds of bacteria, but we recovered, to my surprise, a diverse array of bacteria, even just based on their colors, right? And so after that, the next thing we did was, so all of you guys are present in the corn. Well, it's time to put you to the test. Which one of you can actually prevent or slow down or completely halt the growth of the fungus? And what we did was we set up an experiment where, first of all, we allowed the fungus to begin to grow first. So we give it that head start. And then we introduce the bacteria to see if any of them can actually halt the growth of the fungus, which is really what we're looking for. From that prior step, this one right here, we collected altogether 6,432 consortia of bacteria. That's way too much to screen. And so in order to move forward, we needed some criteria for screening and selecting those that we're going to move forward with. And so that first criteria that we used was the ability to prevent the fungus from growing, okay? And we were looking, this study was repeated twice. And we only moved forward from the study, taking only those that consistently across the two independent repeat that produced, the expected or the desired result. And that is those, as you can see by that yellow box there, we selected wells like those where the bacteria completely prevented the fungus from growing any further. There were instances where you had a gradient here, some they partially allow the fungus to grow. So they didn't halt the growth of the fungus, they just slowed it down. And then you had those, that's the one in the red box here. And then you had those that pretty much didn't prevent the fungus from growing. So it just kept growing. And so what we did was we took those in cases like those contained within the yellow box, and we moved forward with those as candidates that will show promise, okay? What does it say about those that allow the fungus to grow? Does it mean they're useless? I'll come to that in a little bit, but that's not necessarily the case. In any case, we moved forward for now with those that were able to inhibit the fungus from growing, okay? And what we did next was, okay, so you've proven yourself to be able to prevent the fungus from growing. Do you have also, in addition to that, the potential to actually prevent the fungus from making fumonisin? And so what we did was we set up this experiment where we, it's a well-established method. We used it in the previous publications I shared with you. And so in this setup, the fungus will make, a ton of fumonisin, okay? So this was like an ideal setup for us to be able to tell, like, hey, if you can make this fungus stop making fumonisin under this condition that is super ideal for this fungus to make the same fumonisin, then that says a lot about you, okay? The study was replicated. And as you can see in that next picture with the red box around it, okay? Kind of like what I told you, so on that red color you're seeing at the bottom of the tube, that is the color of, one of the bacteria in that mix. And you can see this is kind of red. And so it's turned everything red. The one to the right is kind of yellow. I put these two in particular just to kind of show like the diversity of the bacteria that we pulled out of the corn stalks that we got from the field. Some are green, there's all kinds of color that we pulled out, okay? And so after this experiment, and for sake of time, I'm not gonna go into details of how exactly we did this, but if you want, you can ask me and I'll be more than happy to provide an answer to you. So at the end of that particular experiment, we then measured, well, okay, now it's time to see how much fumonisin the fungus was able to make under this condition, okay? I'll say one thing that I'll probably come back to if it's of any interest to anyone. Take note that in these tubes that you're looking at, the fungus is actually growing. But when we selected the bacteria, it was because they did not allow the fungus to grow here, okay? And so we took the end product of this and we measured for the amount of fumonisin that's present in them. And we took note and recorded the data that I'll share with you in a little bit. The last part of the work then, like I described in the objective earlier in the slides, is then to take the bacteria that showed promise with respect to inhibiting the growth of the fungus and reducing fumonisin production, and trying to find out what genes in this bacteria is actually enabling it or them to do this. So we know exactly the mechanism of action and not just go blind all the way into it, So this next slide is actually a summary of our project status. There's a little bit more further down, but for a start, of the 6,432 bacterial consortia that we started with, we found only 242 that were able to inhibit the growth of the fungus in those plates. So you see that plate on the left with that yellow box there, that's what I'm talking about. 242 of those were able to give us that desired result, okay? Of that 242, we selected a representative, 50 from among them, to then evaluate for that fumonisin activity level that I just talked about. So of the 242, we selected 50 and we moved forward with those to see out of the 50, how many of you can inhibit the fungus or prevent or reduce the ability of the fungus to make fumonisin, okay? And we also preliminarily began to, now that we're getting to a point where we think we are having some promising bacteria, now we want to know, who are you, So that's what this one is. So we sequenced all 50 of them, and within each consortia, there's about 3 bacteria. So altogether, we did for 150 bacteria. And keeping in mind, though, as we moved forward, since we're talking about fumonisin, there are guidelines, which is also part of why this is important. Why do we care how much fumonisin is present in corn? Why do we care what fusarium or the fungus is dumping into our corn? Well, it matters because it's a mycotoxin, basically a toxin, and there are thresholds that have been set by the designated agencies, FDA, USDA, and basically for any corn grain, any corn that's going to be used as grains for food, you can't have anything above 40 parts per billion. So that's equivalent to 4 parts per million, okay? If it's going to be used as feed, you can't have more than 100 parts per million, okay? Now, depending on who's buying from you, what market you're sourcing from, this actually, the range is really from 5,000 to 100,000 parts per billion. So that's basically from 5 to 100 parts per million if you're going to use it as animal feed. And if it's for food, for humans, it goes as low as actually 2 parts per million, and the upper limit is 4 parts per million. This is why we're here, okay? But going on, so again, recall, we're doing this for samples we collected from Texas and Kansas and Colorado, okay? So what I've done is I've kind of broken down this next set of charts by state, just to kind of show you what we found as far as the bacteria we isolated from corn that were collected from each of these states, okay? And from Texas, what we basically found of the 50 that we moved forward with is 8 bacteria. Among them that are from Texas showed promise, and they are indicated by that arrow there, okay? Now, you'll notice a line growing across the bar chart. That's the threshold that has been set, by the agencies as the limit, 4 parts per million. This is for human consumption, okay? It's much higher for animals, okay? So we set that same threshold for ourselves so that we know that whatever we find or what we're selecting, it's a stringent criteria we're applying to it. And really, the criteria we applied is, we're not going to take anything short of 0. Like, I don't want like 0.5, I don't want 1.5. We're looking for, bacteria that will completely knock it down to 0. That's where we want to start from. And from the Kansas isolate, we identified 8 of them that will do that. You'll see the other isolate from Texas here, varying levels of fumonisin that was still possible by the fungus to produce. The limit of detection of this test as of right now is 30 parts per million. So that's basically like 30,000 parts per billion. And for some of them, like this ones, they make actually more than this test can detect right now. New technologies have come out. I think in the very near future, it's already out, we'll be able to test up to 100 parts per million, which is basically the upper limit for animals, 100,000 parts per billion. Okay, but in a nutshell, we found 8 bacterial strains from the Texas collection that were able to meet these very stringent criteria. When we looked at the strains from Kansas, we were able to identify 6. Again, all giving levels of 0 parts per million, okay? And when we looked at the Colorado strains, we found 2 out of the 50 from Colorado that were able to do this. So collectively, we were able to identify from the 50, 13 bacterial strains from these 3 states that have successfully been able to reduce fumonisin production by this fungus to 0 parts per million, okay? And so to kind of update the table I showed you earlier, okay? So we now have these 13 included, and this work has been completed. So all of the stuff I talked about now, this work have all been completed. The work that is ongoing right now is work to then identify the genes within this bacteria that's making it possible. Well, among these 13 different consortia, is it the same kind of genes that's doing it? In which case, well, maybe it doesn't really matter which one, we move forward with, or is it different mechanism that they're using, or a combination of that? That's what we hope to find out within the next 2-3 weeks. But to kind of summarize what I just talked about, when you think about it really, of the bacteria that were able to inhibit the growth of the fungus, it's only a mere fraction of the total bacteria that we have, like 20.7%. And then if you think about those that were then at a later stage able to inhibit the fumonisin production by the fungus, it's a mere fraction of just 5.4% of the total that we have. And so I talked about it earlier, and you can see it on that picture right here to your left of these tubes the fungus is growing. Well, is it possible, one might begin to wonder then that, hey, maybe we've got more gold in this bag than we actually currently realize. Is it possible then that, well, maybe if we skip, the criteria to like, hey, you've got to be able to kill the fungus first before you can be allowed to move forward. Maybe if we skip that and went forward with, let's just find out if we can just, like stop it from making fumonisin. Maybe there's even more than 13 in this pool that we already have. That's kind of just a food for thought, okay? But like I said, we're right now at the phase where we're trying to identify the genes that are responsible for making this. We're super excited. I didn't think, going into this that finding something that would knock it down to 0, or maybe even finding as much as 13, you know, like was something that was going to show up. But here we are. The next phase of the work is to then take this bacteria and actually take them out to the greenhouse and then apply them directly onto corn plants, like we hope you would be able to sometime in the future, near future actually. And then, you know, we'll introduce the fungus there, and then we'll put the bacteria there, and then we'll see, using different methods of introduction, like, you know, how best can we apply it, you know, which one gives the best result, maybe how many applications we need to do to get, you know, like, and then what time is best to put this bacteria in there with the fungus. That's kind of the next phase of the work. And, you know, we're keeping our fingers crossed. So far it's been blowing our minds. We're thankful to God. We've achieved as much as we have, and we're thankful to you for the opportunity to be able to, you know, come this far, participating in, you know, like, in the corn production process, helping you guys do what you do best. And with that, I will stop talking, and I'll thank you again, and I'll be happy to take any questions you might have.
Okay, thank you, Dr. Obasa. It's quite interesting, and I guess in my mind, I was a bit surprised, as it sounded like you were, that you come up with 13 different bacteria to really be looking at in the first phase of this work. We've got time now to take some questions. As a reminder, if you've got some you want to submit and you're watching on the Zoom app, you can do that through the Q&A feature at the bottom of your screen. If you're on the phone, you can just give us a call in our office right quick, 806-763-2676. If we don't get all of our questions answered, if you have some you want, we will share those with Dr. Obasa. We'll include those in the recording that will be posted on our website at texascorn.org. Charles, I'll open up to you if you have a question. If not, I've got some questions that we will relate to Dr. Obasa, but I'll give Charles, our research chairman, first chance.
Dr. Obasa, do you have a an idea how the application will be in the field? Do you have any idea where we're going? How do we apply it? Will it be a liquid? Will it be on a culture medium? Will it be, what will it be?
All right. Thanks, Charles, for your question. That's actually part of the questions we're hoping to address next. Okay. So now we found these candidates. Okay. Keep in mind, you know, like they were in corn. We didn't grab them from the soil. They were already in corn. And so the question is, well, as you know, some of you might be thinking this, if they're already in corn, how come we're not getting the benefit already? Why are we just now recovering this, right? You know, well, those are interesting questions. And, you know, but just keep in mind that, you know, like the fact that something is in corn doesn't mean it reaches everywhere on a corn plant. So it might be in the corn stalk where we got it, but some natural barriers on the plant might prevent it from getting into the cob, which is really where we need it, right? And so it might be that, you know, it's this assistance from our end to then take it from inside and dump it where we need it. Maybe that's all it needs. But to answer your question, Charles, we're going to be looking at different, you know, methods. Okay, we're going to do seed treatment. We're going to do application at different times, and all of them are going to be spray applications. That's one of the benefits of using the bacteria versus the fungus. They are so small, you can put them in your tank mixtures, your tank sprayers, and you can spray them like you normally would, like fungicide, and you know, you'll be able to apply them that way. But we'll in the next phase now be taking this subset and, you know, be looking at different, you know, like application timing and application methods, and see, you know, like which one of them actually, and maybe even number of applications, you know, what combination or set of combinations actually give the best result. And we will report that as soon as we have information on our findings. Does that answer your question?
Yeah, if it's a liquid, you can apply the bacterium directly to the plant surface, soil surface, or whatever. Is there a possibility you could infect a mycelium or a fungus and the fungus carry it in?
So that is basically the idea. Okay. So when I talked about the timing of application, that's one of the things we're going to look at. Okay. Instances where, well, what if you apply before the fungus lands on the plant? Will I still get the benefit if the fungus comes afterwards? What if I apply it about the same time, and put them in there at the same time? Or what if I put it after the fungus already got that head start? Will it be able to play catch up and give me that same result? Those are some of the things we're going to be looking at.
Thank you.
Thank you.
Okay, Charles, do you have any other question before I start with a couple others we have here?
No, I'm sure we'll have plenty of questions down the road, but that's fine. Go ahead.
Yeah, I know, kind of leading into what Charles was asking, this reminds me of work we did years ago. About 20 years ago, we got started into the work with biological controls to help with aflatoxin, and it's not a quick, fast answer. We had over 10 years of projects before we got to EPA with something. So I'd just say, I think we understand from past experience, this is a slow process, but it's one that would be very beneficial.
Yes.
Dr. Obasa, one of the things that come in is, in doing this, have you identified strains that were common across the 3 states, or possibly have you looked, or will you be looking at combinations of strains so one product could be used in multiple different environments, different soil types, whatever that determines this fumonisin to form?
Great question, Charles. David, thank you very much. Yes, we looked at that. So the initial sequencing that we did was to help give us that clue as well, because we also wanted to make sure we were carrying forward as much diversity as we possibly can. But to answer the question as to what strains we might have found that is common across the strains from the 3 states, we did find some that are common. Okay, so these are all gram-negative bacteria. We've got Enterobacter. It's a known strain. It's found everywhere. We've got Panthoear. We have Pseudomonas. So all of these ones that I'm listing are actually strains that we found common to all 3 states. Okay, we have Cosaconea. We have Atlantibacter, and then the last one, the 6th one, that's very common as well, is Serratia. So that Serratia one that I talked about is the one that I showed you that had that red color. So all of the 6 strains we found in combinations and common across all 3 states.
Okay, well, that's positive, something I know we'd like to achieve, and especially since we had partnership funding and starting this project with corn growers from Colorado and Kansas, and to
Right.
national corn growers as well. So that'll be a key component.
Okay.
Charles asked about the application, so I won't go there. That's some things that had been asked. And it sounds like that's a work in progress, which
Yes.
relates back that we'll have to answer. I guess one of the things that we'd have you do is kind of give us, it sounds like some good advancements, really, like I stated, more than I personally would have expected in the first year. So I know you've worked extremely hard on this, but for those listening in today, what would you say are the 1 or 2 big takeaways that our farmers should get from this first year's work, in thinking about this first year's work, and then maybe guidance back to our boards as we determine and work with you on this follow-up work?
All right, excellent question. I would approach this by saying, this work is actually focused on mycotoxin, fumonisin. Unlike, say, a fungus that you've got a fungicide for, or maybe even aflatoxin that's a product for fumonigen, fumonisin is unique and different. There is no fungicide, there is no product you can go out there and spray. So right now, what we're talking about is an issue that currently you don't have a tool to address. And most times you probably end up, dealing with the fallout at the elevator or something. And the other thing that I kind of want to stress, and that I'm excited about going forward. So this is not some, I'm trying to, keep my optimism about this going forward. But one of the things that gets me excited when I think about it is, we're looking at the system right now that, when all the work is done, however long it takes, right, we won't have to worry about, well, will the FDA approve and that kind of stuff? That's not going to be an issue because these are naturally occurring organisms. We're not going to tamper with them in any way. We didn't take them from a frog or a toad in the river and we're moving them into a plant that will then constitute like, oh my God, some GMO. No, this is stuff that's already out there. If you're a producer to deal with, animal people, rare animals and stuff like that, if you sell grains to people like that, I mean, so think about it, right? We pulled all of this out from corn stalk. Well, do the beef cattle already eat corn stalk? Of course they do. Do we know of any consequence from that? No. So what I'm essentially trying to say is we're starting from a point of advantage to try to address a problem that currently right now, there is no tool right there out there to help address that. And I hope going forward, working with you, with your support, I hope we can get to the very end of this. And, there's a whole bunch you're contending with right now, market volatility, weather, and all of that. If we can just take one thing out of it, especially something that can, impact, the rated value of your corn crop, that would be huge and significant. And I hope we can see this through. I hope that kind of addresses the question.
Yeah, you hit on something that we've worked on a lot through our board and a lot of our other checkoff boards and with products is meeting guidelines in order to get through approval through FDA, ultimately through EPA, and having this naturally occurring native to the corn plant will be a big asset in speeding that process.
Yes.
I would not want to lead anyone to think that it will be fast and easy, but it will be much easier having that native to the corn plant thing hanging there with us, and the fact that we're already, everything that you're going to be doing is already exposed to the environment, to the use of the corn.
Yes.
So, well, Dr. Obasa, we're nearing the time we need to end our conference.
Okay.
We'll take any further questions that people have. We'll pass them on to you. We'll get answers. And then this will be posted on our website at www.texascorn.org. Thank you, Dr. Obasa, for giving us the update. Sounds like some very good work on a project that certainly at times has cost some of our producers significant monies. It cost crop insurance some significant monies with quality loss, and something, if we can get an answer, it certainly will help with the market stability for our producers. Thank you.
Thank you. I appreciate the opportunity. Thank you all.
Okay. Thank you all for listening in today. That's something we hope to be doing more of in the future. Have a great day and keep putting that seed in the ground.
Thank you.